research brainstorming session. We have two infectious disease topics today. First, Dr. Belshan and then Dr. Velagapudi are going to give us brief presentations with opportunities for input, questions, comments. These sessions are meant to stimulate collaborations and interactions and expose some of our learners to the research we're doing to provide opportunities and education, and also, you know, strengthen the quality of our research also altogether by putting all our heads together as we think about the research questions under study. So thank you both to both our presenters today. And one last thing is our next session is Friday, May 26th on epilepsy, neurology, and Dr. Herman and Dr. Simeone, a pair of basic science clinical faculty investigators, will talk. I think so, and it says a reminder down there if you'd like a different email to make sure you get this communication for upcoming sessions. Throw that in the chat, and I think that's everything we need to talk about up front. So I will turn it over to Dr. Belshan.
All right. Hopefully. Oh, can I get my screen?
Yep. Oh, there you go.
All right. All right, is it being shown?
Yeah.
Okay. So yeah, I wasn't sure exactly what to do for this. Well, except I will talk briefly about where my laboratory is going right now, but I thought I'd give a brief introduction to kind of what my lab has done throughout my history here at Creighton. I came here in 2005 from Washington University. And at that time, I guess basically my whole career, I've basically been sort of a molecular virologist, preclinical, and most of my studies have focused mostly on host virus interactions at the cellular molecular level, and kind of an interesting history to always go back and reflect on, from the sense that you know, I started out working at nuclear entry and pre-integration complex, basically looking at basic aspects of HIV replication, and looking at, you know, mostly, like I said, cellular factors that mediate things. Because of course, you know, the drug companies, big pharma's always been interested in developing HIV inhibitors targeted towards HIV enzymes. So there's no sense doing that. And so we were focused mostly on cellular stuff. And then slowly I kind of evolved into getting into proteomics. And in about 2008, and I've done a massive amount of proteomics, and I kind of have a love-hate now with proteomics after everything is said and done. And that really I've done up until pre-COVID. And most of, like I said, most of those studies are basically on virus replication, host cell interactions. And then right around 2012, NIH kind of basically stopped really wanting to fund basic research. And they became just mostly interested in what they called cure research, which was kind of trying to disrupt latency in infected people. And so I kind of switched to that and was ramping that up. And I'll talk a little bit about that here in a second. And then of course, COVID hit. And when COVID hit, I decided to just sort of switch what we were doing and work on that. And basically in the last 3 years, we've done mostly a lot of sequencing of COVID, doing a lot of evolutionary analysis, some virus stuff, not as much as I probably would have liked to have done. I'm not a genetic and I'm not an evolution guy, but that's basically kind of what we got into, because we ended up getting a big contract with the state to do some of the surveillance sequencing for COVID through the DHHS at that time. And then, yeah, and so now that's kind of ramped down. NIH is also not interested really in any sort of virology with SARS-CoV-2 either. So it's kind of back to HIV, I guess, and kind of what I'm going to talk about here in the next 10 minutes or so is these, you know, what we're trying to work on or work our way up to now going on. Which is, and then in general, the other thing I always like to put out there that I've done in the past I've done a lot of antiviral drug testing for various, interacting with other people. We have that capability. People occasionally come to me and say, hey, I've got these drugs that I think might inhibit, RT or integrase or something. And we've done that over time. And then I saw Chris is on the call actually over the last decade and a half even. I've worked with Chris a little bit here and there, helping him test nanoparticle formulations. He's been really the driver of that. I've been kind of just the guy with the virus a lot of times with that. But I'm very thankful because Chris has done a lot of great work with that, and he's allowed me to help him with that, which has been good. And then a couple other oddities I've worked on. I've worked with some people in arts and sciences on looking at susceptibility with exercise, as you can see. And then also during COVID, Steve Gross and Steve is I think he's in dentistry, but he's also in chemistry. He wanted to work on developing some 3D resins conveying antiviral properties. And we actually worked on that. And it turns out successful, but I don't know how successful it was because people don't care about COVID anymore. And so I don't know, he always mentioned that he wanted to try to shop this to companies, and I don't know where he is with that, but that's kind of my lab and where we've been in the last few years. So now as we're transitioning back to HIV at this time, and sort of there's two things that we're really working on right now. One goes back to sort of, host cell interactions, and that's looking at susceptibility, and I'll briefly talk about that here. And then the other one is looking at persistence and latency. So I'm going to prologue this because this sort of comes off of the study of susceptibility, comes off of another project that we were working on, which was looking at host cell factor cyclophilin B, which is I always call it the ugly stepbrother of cyclophilin A, which is quite well known HIV factor. It's in the immunophilin family that's thought to be an ER chaperone. The weird thing about this protein for us was it kept popping up in almost all the proteomic studies we did. And so we became interested and we started looking at it, and what we could find, or what we found initially, was that, and it's highlighted down here in this publication, that when we overexpress cyclophilin B, it actually seemed to enhance HIV replication. So we asked the question then, of course, if we knock it out with CRISPR or RNAi, is it necessary for HIV replication? Turned out it was not at all. And so that kind of killed that project. But as part of that, we were basically trying to make a bunch of cyclophilin knockout cells. And this led to this sort of interesting story of what we're doing right now. So what we did is, in the process, we would basically use cell lines and we'd try to knock out cyclophilin B. And then we would basically clonally isolate the cells. Some cases we did, and we kept bulk cells. And then we would test them for susceptibility to HIV. And then, we would we do a primary screen. Those were done in bulk and like 96-well plates, lots of cells, hundreds and hundreds, as you'll see in a second. And then we would secondarily confirm them. And then some of them, if they were still CD4 positive, we would actually test for replication. Like I said, we screened, and this is just a subset of those cells. We screened a lot of different cell lines. And what we found that was interesting was the susceptibility was all over the place. So we could find cell lines that were completely enhanced for infection, cell lines that were refractory to infection. And then the oddity of all of these things was that, especially on this slide, only like, 2 of the cell lines on the slide were actually cyclophilin B negative. So it didn't have something to do with that. It likely has something to do with either purifying a certain kind of cell or an off-target CRISPR effect. And so we also did this in other cell lines. We have HeLa cell lines that we knocked out. We have THP1 cell lines that we knocked out. 293T. We have a whole bunch of different models that we're working with, but we're working basically now with the surrogate T cells because we can actually do replication assays. And I'll just point to this one on the right, which is basically we have cell lines where basically if you try to replicate HIV, normally it goes, you'll start with a very low MOI, and after about 10 days it'll take off. And of course, it usually eventually will kill this tissue culture out. But then now we have cell lines that seem to be really refractory in different cases. And so the question that we're left with is actually why? And in the hope that we could actually find a cell factor that really is necessary for HIV infection or efficient infection, or or inhibitory to HIV infection. And so what we're doing actually right now is doing some RNA sequencing on some of these. These are some other lines. We actually reconfirmed them after we took them out of the freezer for 5 years or so. Reconfirmed some of the phenotypes. These are different Jurkat cell clones, and we're actually doing an RNA-seq right now to be able to do that. And then, of course, after that, we'll do all the sort of validation studies, host cell studies, where we basically either do gain of function or loss of function studies, that type of thing, to try to identify whatever these novel factors are that are changing or altering the cell's ability to do susceptibility. So this is the long shot one, that this project that could potentially lead to something, I hope. But again, it just depends on how well the RNA sequencing goes and that type of thing. So the other project that's more germane to what NIH is interested in right now is actually looking at these mechanisms of HIV persistence. And so, as many people know, right, we have very effective therapies for HIV, and patients can get undetectable levels of virus, but but everybody still, all those individuals, they don't become cured, and if you take off their therapy, usually they'll rebound pretty much immediately after cessation of therapy. And so and the reason for that is because basically, as studies have shown, that basically even in these well-treated individuals, there basically are infected cells in every tissue in their body. And the key thing that is kind of interesting to me that all along was, originally they focused mostly on latency, which were cells that they thought were basically hiding in the body. But more recently, it's become much more accepted that HIV, that there are actually still even persistently replicating cells or persistently expressing cells in the body. And both of these actually kind of comprise this persistent reservoir overall. And so, it's beyond T cells, probably macrophages, microglial cells, astrocytes. There have been multiple studies showing multiple different kinds of cells can actually be infected in the body. The other issue is, even beyond these types of cells that are these viable cells, there are a lot, they've shown that there are many cells, T cells in the body and other cells, that will actually harbor defective viruses that may only, express one or two HIV genes. They can't make replication competent virus. But the overall problem in that case with the persistently infected cells is that by making HIV antigens over time, especially things like TAT, which is the activation regulatory protein, people basically end up getting, chronic immune stimulation, chronic inflammation, that type of thing. And that's one of the contributing factors to the disease state, the long-term sort of long-term disease state of these individuals, is this chronic inflammation. So the NIH actually just put out an RFA earlier this year, actually to basically address this mechanism, to try to look at chronic infection and chronic inflammation in individuals. So as I said, we already had these models of latency from some previous studies. I'll just kind of briefly go kind of over this. So what we wanted to do is to model latencies. We wanted to use cell line models, and the reason why we want to use cell line models is because they're much more controlled, usually more uniform, although we can make heterogeneous lines. And so the model of latency that we use is based on a virus called the dual flow virus, which is interesting. It's an HIV clone where they knocked out envelope, and then they put in a GFP reporter for active infection, and then they also inserted an M-cherry fluorescent protein under the elongation factor 1 alpha promoter. So what this virus does that's kind of interesting is in actively infected cells, it will be both green and red. So ergo, it will be yellow essentially. But if the cells are latent, it will only express M-cherry. And we basically, you can see this kind of on this flow cytometry plots of this with M-cherry on the Y and EGFP on the X. And when we infect cells, you get something like this, where you get the bulk of them will be dual positive actively infected cells. There'll be some latent cells, and then of course uninfected cells. So we went through and isolated a whole bunch of different cell lines, either by flow or limiting dilution, where we basically would sort of gate on either the active or the chronic population, and either sort them by flow or do limiting cell dilution and look at them that way. And basically we have multiple different cell lines that are either latent or actively or chronically infected, or have sort of a mixed phenotype. overall. And these actually are, you know, I think are a good model to be able to kind of look at and attack, you know, HIV persistence. And the other thing I'll say, the one thing I haven't mentioned about these that's interesting is on our end, you know, one of the big questions in the field is, so if these cells are infected and they're chronically infected, usually most of the time cells will die. And so the question is if you can make cells that are chronically infected, how are they able to survive? And that's one of the big questions that we want to ask overall. So when we model these also, so this is kind of a different format to look graphically kind of at the numbers, but I'm going to jump around this a little bit because I'll first mention, so, you know, we have these numerous different chronically infected cells where, you know, and the yellow bars are the actively expressing cells. They express both GFP and mCherry, and we have a bunch of those. Then we have a bunch of latent cells, and we always check the latent cells to make sure they can reactivate. And there are a number of different latency reactivation agents that are available. The biggest, usually the strongest one is PMA with ionomycin over here. And you can kind of see these are just some of the subsets we have. And the reason why we do this is because, for example, this JDF2 line stands for like Jirga dual flow, which doesn't reactivate at all. It actually has a defective provirus in it. But then we get other ones, like I said, that work really nice, that are really mostly latent, almost upwards of 98%, 99%, but they're reactivatable. So I think this is the last slide I have. So really, as I'm thinking about putting this together, and this is something I'm putting together right now, is kind of trying to look at, use this model to try to define, number one, to define the mechanisms of HIV-induced persistence. And basically, we're going to go back to what I was talking about before and basically use RNA sequencing of these different types of cells to try to see what's different in these cells. Why can they maintain HIV in this state overall? And then the other thing that I'm actually excited to get into, but I actually know very little about, is this using locked nucleic acid technology, and maybe somebody out there knows more about this. If you do, please let me know. But basically, this is an interesting technology that was, I think it was discovered back as early as the 2000s, where you basically lock, I think it's the 2' oxygen, or is it 4' oxygen? I cannot remember my chemistry, but you basically lock the base, the ribose base, in either DNA or RNA. And the effect of this is actually that it creates a more stable molecule, especially, and it also creates a much more stable duplex when the nucleic acid duplex forms. Like they say, like the estimate I read was for each of these bases in an oligo, it would increase the binding, the TM temperature like 10 degrees. And so HIV, so the idea for this is to actually create RNAs or RNA oligos with locked nucleic acids, and actually use them to target stem loop structures in the HIV genome to basically disrupt replication or reactivation of the virus from latency. And so I'm gonna try to do first is design some of these to a couple different well-known elements in the HIV genome, including the TAR element, which is necessary for transcription, the GAG-Paul frame shifting site, which is necessary for expression of the enzymes of the virus, and then also for the Rev responsive element of the virus, which is necessary for envelope expression and GAG-Paul expression. But that's another one that just is in its infancy that we're trying to do, but I'm actually kind of really excited. The reason why I got excited about this was there was actually a SARS-CoV-2, well, it was an influenza A and SARS-CoV-2 paper that was in Nature Medicine, where they were able to target and show knockdown, and they also use these in animals in an in vivo model to show that they could actually use these in vivo. And nobody's done this actually with HIV yet. So that's kind of the cool point there. And that would be the hope, would be able to sort of develop this, show it if it works, you know, in tissue culture, and then move from that to animals to other things. And that's kind of all I have. Hopefully that's good enough.
Great. Thanks, Mike.
Mm-hmm.
You know, I'm going to start because that's, I have the option as the moderator to ask the first question while everybody's gathering their thoughts. So when I look at your aims, I mean, it's really interesting, but aim 1 is looking at like host cell responses, right, to HIV infection that determine latency or active infection, right? And AIM-2 is looking at like impacting the virus itself, right, to reduce its infectivity or effects on cells, I guess. So I don't know if there's a way to tie those two more closely together when I look at it and think about it as a grant proposal, rather than like two really very interesting and promising projects. And when I look at your AIM-1 with the RNA-seq, what I think would be really interesting, and I don't know, maybe this is not possible given, I don't know what these cells are, and given your JURCAT data with the HIV, work where you did the CRISPR targeting and you got really unexpected results. I don't know where that variation comes from, if that comes from the parental cells or if that's a CRISPR outcome. And so I don't know if this is possible, but it seems like doing the RNA-seq in combination with a CRISPR screen where you've got this nice flow and sorting assay for latent and active infection. So if you did put a library of CRISPR guides in to target, a genome-wide library, or, more targeted toward likely suspects, maybe coming out of your RNA-seq, and then do your CRISPR screen, and then however long you need to sort for, like, if you have upregulation or downregulation of these genes, now you don't have an active infection anymore, or you don't have you can switch the characteristics of the population in that way. And that would get you to, we all know the difficulty with RNA-seq is you get all this data and how do you make sense of it and where you go. But this would be a way of really like taking that information and going in a really focused mechanistic direction. Maybe that's not possible with the cells given your first part of your talk, but I don't know.
Was it actually, interestingly enough, I tried that. What was that, about 6 years ago? I have the whole CRISPR genome I got through AdGene, one of those original CRISPR genomes, and we tried treating cells and then infecting them, and we did them in parallel. And I think what you're saying now probably would work with like single cell RNA-seq. But when we did that, we were trying to look for bulk differences if we hit cells, and the issue is we can't see it, and so I was always hesitant to follow up on it. Exactly, kind of what you're saying, but I wonder now the technology might be good enough to be able to do that.
Well, we have Dr. Fu on the conversation, who's an expert in our next generation sequencing techniques and the director of our innovative genomics core. But I think you do a bulk seq, but you look for I guess dropout or overrepresentation of particular genes with the guide barcoding. And since, you could sort them after a period of time to allow for selection of, cells that are moving into the persistent or the latent or acute or latent, whatever population, and then do your bulk seq on those and look for what guides are popping out, showing you that there's a mechanistic, like that they're drivers of one phenotype versus another. Lucy, I'm describing it poorly, but I'm sure you have better language than I do.
Yeah, so I think for RNA-seq, it's more will be on a
RNA
level.
DNA-seq, not RNA-seq.
Oh, if you are trying to do the CRISPR screening,
with the
yeah,
DNA.
it will be more on which gene will be important to regulate that. But if you are trying to find something from the RNA-seq, it will be more on higher expression and low expression will affect it.
Sorry, I meant, I don't, that's why it's great you're here. The DNA with the CRISPR screen. to look at whether targeting in a global way, targeting particular genes can push your cells to a different phenotype. Thank you, UC. I don't know why I can't keep that straight.
And there's a question in the chat too that just popped up. Doctor, is there any lab that has a model where post-ART there is no reactivation at all?
I don't think so. I mean, because that would essentially be curing. And I know, there's been sporadic reports of people who've created mouse models where they've been able to cure mice with human infected cells. But as far as I know, no, there isn't a model for where there's no reactivation. There's always some level of reactivation, although I will say in most models of latency, the reactivation amount is very, very small that they can actually reactivate.
So I wonder about your persistent latent versus active. And you really want to like push them to a third state, which is no virus.
So that's not an area I've gone into. I know that NIH, there are people out there looking at, and again, it's mostly CRISPR targeting of the HIV genome that's been in development. I suspect it's not working very well for people because I think people have been trying to do it now for over a decade. One of the things, and this is why I'm also interested in the locked nucleic acids, is, initially with latency, the idea was they thought in patients that they could treat patients with a reactivation agent. And they actually did this with a couple of them where they would activate the virus in the hopes that if they were under treatment, their immune systems were functional, that you would activate these cells and then the patient's immune system would see them now finally and kill them, sort of what they call the shock and kill method of latency. And they've tried that and it's failed. And so now they are looking for methods. This is another push for them, is to look for methods, what they call shock and lock, where you actually where even if the virus is activated, it's locked down and can't express. And that's kind of where I was trying to go with the locked nucleic acids. Because if you can add a drug in there that will stop them from expressing, that would be perfect. But again, it still won't cure them because of the integration. I mean, that's just the whole kind of end game for why HIV can't be cured, is there's always going to be some cells that have integrated virus.
So you really do want to drive cell death in the infected cells. That's the bottom line.
And that's that push of the RFA that they came out with, that they want to figure out a way. They want to mechanistically know, how do these cells survive, and then can you exploit that somehow to get them to be more, I guess, susceptible to killing.
You could do that with a CRISPR screen too. I think that's what I would try to do with your AIM-1. Any other comments, questions? All right, thank you very much, Dr. Belshan. It's time now to turn to our second presentation by Dr. Velagapudi. Thanks so much for being here, and you should be able to share your screen.
Thank you, Dr. Hans. That was a great presentation, Dr. Belshan. I have a tough one to follow. Mine would be very much of clinical research. I will try to keep you guys engaged. Yes. So I started the HIV and PrEP clinic at Bergen Mercy, and treating HIV is my passion. And so I have a couple of projects which I already published, which is about studying readmission patterns in people with HIV and hypertensive emergency, and clinical features and outcomes of COVID-19 in people with HIV. And the third one is just a review article on cytomegalovirus encephalitis in HIV patients. The ones in progress are also again more clinically relevant questions or something we see every day in practice. So the first one is studying the statin prescribing practices in compliance with AHA guidelines in people with HIV. And then the second one is studying the, actually I have to edit the title. It's studying the readmission patterns in patients with HIV and Burkitt's lymphoma, which is a AIDS-associated malignancy. Studying the readmission patterns and the predictors of readmission in this population. And the third one, which we are almost close to finalize the manuscript, is studying the predictors of COVID-19 vaccine acceptance in people with HIV. This we include, and I'll tell you more about the study, but that's almost close to be done. This is the one that is really in progress. Studying the statin use, prescription practices, and to make sure their people with HIV are on the right dose of statins. So the primary objective is to study those practices, and the idea came because even in well-controlled HIV, it is a pro-inflammatory state, and 2 out of 3 deaths in people with HIV are secondary to cardiovascular disease because the currently available antiretroviral therapy is so effective that persons with HIV live as long as their peers. So they die of something what a non-HIV person dies too. So it's cardiovascular disease, cancer. These are the ones, these are becoming the main cause of death in persons with HIV too. And there are multiple studies that have shown that statins are useful in knocking off that pro-inflammatory state. So this would be a study we would be doing in at VA Omaha. So the veterans cared at VA Omaha are the ones who would be included in the study, and whoever has a visit in the past 1 year is who we are including. And we'll be calculating the ASCVD risk score, which is a calculator based on age, race, gender, and whether they are on any medication for hypertension, are they smokers or not, and do they have diabetes. Those are the main ones that come into the ASCVD calculator. And the patients who have contraindications to take statins, it can be allergy, it can be intolerance, or it can be drug-drug interactions. Those would be excluded because even if they met the criteria, they can be on the statin because of these reasons. Or patients who, so we'll identify this by chart, the study population by chart review, and look if they come under, categorize them into low risk, moderate, or high risk, and see whether they are on the statin or not. So primary outcome is to study the statin prescribing practices. Secondary outcome is to look at the dose, if they are on the statin, if they are on the right dose. And if they are not on the right dose, the treatment team would be alerted through the VA EMR, which is CPRS, and those changes would be made. That's one of the project that is in progress. And the other one, this one, we're almost close to be done. So we looked at the COVID vaccine acceptance in persons with HIV. And we found that non-smokers and those with CD4 count less than 200 in the past 2 years, and those who received influenza vaccine in the past 5 years were more likely to get the COVID vaccine. So it's a simple retrospective chart review, and we are close to finalizing the abstract too. And Dr. Distach is my research mentor who guides me on all these projects. Sorry. And the last one is studying the clinical outcomes and readmission patterns of HIV positive patients with Burkitt's lymphoma. This we would be using either NIS or NRD database. to look at these readmission patterns and then stratify by their comorbidities, who are more likely to be readmitted, and also look at the clinical outcomes like length of stay and in-hospital mortality in this group of patients. So these are the 3 main projects that are, excuse me, still in progress, and that's all I have today.
Well, is it open for questions, Laura?
It is, sorry, it took me a minute to get to my microphone. Please, Maureen.
Well, Manasa, I always love hearing about your research, not only because I love the questions, but I also love to hear the passion in your voice about it. And I think that's one thing you share with Dr. Belshan is that passion. And so one question I wanted to have, when you were going through your slide about the the predictors of COVID-19 vaccine acceptance, is an issue near and dear to my heart. Did you say that smokers?
Non-smokers, sorry.
Okay, which what one expect. So in going through this, did you compare the level of your overall population? Did you compare them at all to the general population? So were people with HIV more or less likely in comparison to the general population to get COVID-19 vaccine, or was that not something that you looked at?
They're pretty much close, Dr. Tierney. I mean, if we go by, I looked at the Midwestern, COVID vaccine acceptance in the Midwest cohort. It's close to 51% is in our institution in the study and the population we studied, and it's close to what it was in the
General population.
general population in Midwest too.
So it's only in those people who have faith in vaccines in general, who understand that they're more immunocompromised because their CD4 count is less than 200 and follow healthy habits as non-smokers, who had higher acceptance, right?
Yeah.
I'm actually surprised it's not higher in the overall HIV population, but
And I have to
do you have any thoughts about that? About why?
Actually, I have to clarify. It was 51% was the booster acceptance. I have to look at the primary series. Yes, it was higher. This was the for booster. 51% was for the booster. It was higher that we didn't. I don't think we calculated the overall percentage, right, Dr. Distach? Did we? We didn't look at the overall percentage, I believe.
Oh, no, we didn't look at the overall percentage.
Yeah, but that's a great thought, Dr. T. And we look at the overall percentage. For some reason, we looked at how many were interested in the booster, but you're right. But if I have to guess, it was definitely higher in this population than, slightly higher than the general population. Booster acceptance was similar.
Okay. Thank you.
Kind of surprising. I mean, those people are, that population would be at much higher risk for severe COVID, right?
Yes. Yeah.
I wonder if the message got to them or.
So primary vaccination, most of them got it, Dr. Hansen. So primary vaccination acceptance was much higher. By the time of the booster, because I documented the reason for, I collected the reasons for refusal too. Most of them were like, doctor, you told me to, I did my primary, don't ask me about my booster. Or if they are a little farther in the pandemic, they're like, see, I didn't get it till now. I'm taking my pills, I'm undetected. Why do you think I'm at increased risk than others? Was most of the time, most of the reasons for refusal were like, they were like, we are tired of this. We just got the primary series, that's it. And the second most common reason was there is so much of vaccine hesitancy. they didn't believe that it is true that the pandemic happened, and they thought it is exaggerated than what it was. Yeah. So there was so much of vaccine hesitancy, especially with the boosters. Primary vaccination, yes, they were more receptive, but after that, or they would say, well, I will get it at my pharmacy, not during the visit today, but they come for the next visit and it's still not done.
80% vaccine acceptance overall.
Which is definitely higher than the, yeah. Thank you, Dr. Distin.
Interesting. I wonder if the two of you, Dr. Belshan and Dr. Velagapudi, if you, I don't know, see any areas of intersection or collaboration, or like questions that should be asked on one side or the other that come to mind.
My interest is a little piqued with the statin study, just from the standpoint that I assume I assume, and again, I don't know much about the clinical features of long-term HIV infection, but I assume that people, as she had on there, have an increased risk of cardiovascular disease, and I imagine that ties into the chronic inflammation issue with patients, and it might be somewhere where we could find some common ground. Since that's what I'm trying to look at now, but of course, like I said, it's nice to know that you're interested in that, because of the fact that I usually deal with in vitro models and not in vivo, but NIH is always more interested, of course, in vivo studies and things of that nature. So
has the inflammation been characterized?
You mean markers, Dr. Townley, or?
Yeah, like SED rates, CRPs.
So CRP, there are some papers that said CRP is high in these patients, but they were limited by the sample size and other. It's not an established fact what exactly is the surrogate inflammatory marker for these. And there are some molecular level markers, which I don't remember, but CRP was studied, which was equivocal.
And I guess could you look at coronary CT scores, coronary calcium CT scores? Because that's also another test to help guide you in recommending or not recommending a statin. And my third question would be, could you look at creating an ASCVD risk score that would include HIV? To figure out, well, I guess if you have HIV, you should be on a statin, or should you not be on it? I mean, is it related to family history and all those other things that you look at when you look at lipids?
Yeah, so your second question, whether coronary calcium scores, yes, that's something I was thinking after the most recent grand rounds in Department of Medicine. So coronary calcium score is something I want to offer for my patients, but I believe it is still, insurance would pay only if they are above 40 or 45. I don't think I can do it on the younger ones, but whatever is the insurance approved age limit, yes, I can do that and use that as a marker for statin prescribing practices. And the third question you had, so HIV status is not a part of the ASCVD scoring, but we just do it like how we do it in the non-HIV population, the same calculator on the AHA website. But that might be something interesting to look at. And ASCVD score also, it is not validated in younger patients. So the younger patients, honestly doesn't have any marker. neither CT coronary calcium score or ASCVD scoring, but above 40 or above 45 is where all these tools can be used. I can look more into if there is any specific scoring that is that can be used and takes into account the HIV status.
Yeah, those were just some thoughts I had, so.
Do statins reduce inflammation?
Yes, Dr. Hansen. Yeah, they do.
And I guess risk factors for heart disease, etc., like are a lot associated with increased inflammation. So it would seem like, I mean, I'm out of my element, but it would seem like people with HIV are at higher risk for
Yeah.
heart disease because of the, so that's kind of interesting if that's an intersection of what you're interested also, Dr. Belshan.
Yeah.
If there are, I don't know, markers that might be looked at that could give clues to the, you know, the preclinical research.
Or like studying the markers,
Yeah,
to
study
measure the
markers or,
inflammatory state. Yeah,
yeah, the causes of the chronic inflammation that links the two diseases and how you could mitigate that, I guess, in HIV patients in additional ways.
yeah. Yeah, that might, yes, I agree with Dr. Belshan's comments and your comments. Yeah, inflammation is something we might be of the interest for preclinical research. And Dr. Belshan, you said you are doing more in animal models, not the patient samples or anything, right?
No, not even animal models usually. I am very, very preclinical. Not that I have an aversion to animal models. I just have never gotten them going. I know Chris at one point did, but then he even went to a collaborator. I do know people who do animal models, but I don't know if animal models are necessarily relevant in this case, because a lot of times basically what they do is take an immune compromised mouse and grow human cells into it. And so it's not, like you said, it'd be interesting. It's something to think of quite a bit about how to model inflammation. That's the biggest issue because of all the different spectra of factors. You know, I don't even know is it really clearly understood exactly what drives the chronic inflammation. I mean, I know there are different factors, but it's definitely a big ball of yarn to try to untangle, and it does give me a lot of thoughts, but kind of can't think of a good lead yet.
Got you.
I wonder if you had in your, like in your RNA-seq that's to come, if you know, looked at inflammation pathways, then you could go to some patient samples and try to validate a correlation between these markers, whether they're inflammatory or others, on, you know, latent versus active infection, etc.
No, absolutely. No, one thing that would be very helpful for me, especially for NIH submissions, is, you know, if we have a cohort of patients to get samples from, because they always want everything now to be in tissues and human tissues of patients and that type of thing.
I think it'd be fun to toss questions around, you know, from the basic science side and the clinical perspective and see, I don't know, what answers or how you refine the questions and the, you know, whether there might be some collaborative possibilities. That would be pretty exciting.
Sure.
Yeah, that was one of the questions I had was, so in these patients, do you have like blood samples?
No, Doc. We don't have any blood samples. This is a chart review study.
Okay.
looking at the veterans chart to see whether they are on the statin or not, and we calculate the score and see if they needed a statin. If they are not on a statin, we'll communicate with the team to get them started on the statin.
But most of these patients you do see like in a clinic, so like
Yes,
going forward, it would be possible to get like blood samples. Because like you can do inflammatory assays that are very detailed if you just spin off the plasma. And then, run it on the plasma, and then from that you can get, often now there are kits that give you like 54 or greater different markers, and then you can look at those independently and correlate them with, various things.
Yeah. So I have to look more into what were the already studied markers or markers people might have thought that would be really reflecting the inflammatory state. And I think then we have to figure out how to do those tests without patient getting a charge. That's another thing in the clinical world, because this would be purely for research purpose. So those are some things look into.
Yeah, I think that would be, some kind of small grant proposal to fund those kinds of studies, and probably more conversation certainly than we have time for today to refine a hypothesis that might, generate some preliminary data that Dr. Belshan, you're putting together NIH proposals that, you get some preliminary data, and then you can build that into a proposal to ask for bigger dollars for a more expanded study.
Yeah, no, that would be the ideal plan to.
Yeah. All right.
This is Giri Andakuri. I just wanted to just add on to that. I think it's important to find out the differences between chronic inflammatory states and how HIV has come to be defined as such, just like, rheumatoid arthritis and any kind of chronic infection. Is it similar to that, or is it different? And actually, I've learned a lot from this presentation, these presentations today, I should say. The latent infection, does that kind of express any kind of inflammatory markers by itself in patients who have this latent infection or the cells, if you will? I think that'll be even more specific to HIV, and it'll be, again, going back to Dr. Townley's point, at some point add a marker and a point in the risk scoring system, saying that if you have HIV, this is what you express, hence you're at a higher risk for cardiovascular disease. So there's two-step process. One is to see the expression of any kind of markers in latent HIV patients, because most of these patients are being treated, and it's important to understand that they may not have active infection, ongoing infection, but they do have latent infection once they are on treatment, and see what the markers are, and try to measure them and make sure that they also add to the cardiac risk in those specific patients. Because just to know somebody has chronic inflammation and not knowing what the forward effect is, it'll be nice to establish that. I think there are lots of clinical studies establishing that, hence we follow it. But I don't know if there's any cell line based or animal based or any kind of translational research on that. So that's very cool, very interesting. Thank you both for the great presentations. Just a thought.
Thank you, Dr. Andakuri. And looks like there is a comment in the thought.
I had a question. This is Julie Fairbeck. I'm one of the ID fellows and working with Dr. Manasa on the HIV and statin project. For the internists in the group or primary care, are you guys ordering the coronary calcium score? Are you ordering that for any of your patients, like just in general, not HIV patients? I did internal medicine for a long time, and I don't think I ever ordered it, but I think it's kind of come and gone somewhat, but I don't really know where it stands right now in like primary care practice, if primary care doctors are ordering it, or if it's really just the cardiologists that are ordering it.
As a primary care, I order it mainly in my patients that I don't know if they need a statin or not. So, I'm not going to order it in a diabetic. They already need a statin regardless of what their LDL is. But, if somebody, 50, 60-ish-year-old female with, a high statin and no family history, what do? It kind of helps guide that risk assessment. I have not done it in an HIV patient, and as the younger you get, the less it's not good in anybody under 35 for sure. So, which may be a fair amount of the HIV patients you're seeing.
All right. Well, interesting. It's always a surprise where we go when we have our clinical faculty and basic science faculty together. I think that's really fun. And thank you both for really, really interesting and really great talks. So, and for the audience for being here. And we will see you all hopefully next month for a different topic of research. Good afternoon.
Good afternoon. Thank you, everyone.
Yeah, thank you.
Thank you. Good afternoon.